All right, good evening, everyone. Thanks so much for coming. Welcome to Science in the Club. Take a seat, get ready. There's a lot of you here. This might be a record attendance for the year. It is good. Well, it's good because it's our final show for the year. Since we've got so many people, can I have a show of hands? Who's here? Is this their first Science in the Club? All right, good. Lots of newcomers. All right. So I should warn you, this is going to be an intense night. This is going to be very science heavy. Shut up. Don't ask questions. No, I'm kidding. This is supposed to be fun. We have some real hardcore scientists here, but the whole point is for these events to give you the opportunity to hear about science from these people themselves. Now, my name's James. I'm going to be sort of wrangling our scientists and making sure they behave, but hopefully I'll be doing very little talking. We've got 3 speakers here tonight. We're going to hear from each of those. We're going to do some science trivia to test you on how well you were listening to the talks. And then we're going to take a bit of a break and we're going to have a Q&A at the end. So as you're listening to the talks, if you have any questions, write them down or remember them, and we'll bring them all up at the end and you can ask them their questions. And these events are really great because we're in this golden age of science communication where there's so many documentaries out there and popular science magazines and things, but at these sorts of events you get to hear the science from the minds of the people that are actually making these discoveries. And not only do you hear about the science, but you hear about the stupid stories about how this science happens and the mistakes that they make and the crazy ways that they come up with ideas. And that's what makes these nights really special. And the 3 speakers we have here tonight are the walking embodiment of a Slim Dusty lyric in that they are looking forward by looking back. That was a really funny joke for the kids in the audience. Slim Dusty, look him up, kind of a big deal. Unlike that other Slim Dusty song, this is a pub that definitely has beer. So grab a drink, take it easy. And we're going to hear from some UNE paleoscientists that are looking back into the past at how plants and animals changed over time. And this sort of information is becoming really important because it's enabling us to understand how we can then deal with the changes that are happening in the environment in our own lifetime. But I'm going to stop talking and hand it over to our first talker. He is Dr. Professor Lord of all Wonder, Stephen Rowe. Give a big round of applause for Stephen. Come on up.
Thanks, James. My name's Steve, and it's been 3 weeks since my last drink. Sorry. Oh, crap, wrong talk. That would have gone over the kids' heads, I hope. Now, I sort of run a lab called the FEAR Lab at UNE, the Function, Evolution and Anatomy Research Lab. We look at shape and we look at using computers to model mechanical behaviour. And we do other stuff too. Most of what we do is paleo related. So most of the projects people in my lab are involved with include some sexy extinct fossil animals, but not necessarily all of them. Some are entirely zoology based and some are biomedical. So looking at questions with respect to surgery, etc. Now, where am I supposed to be clicking this?
Big button.
It's high tech, huh? Right. So there's a bunch of people in the lab. This is a lot of them. Initially I was going to go through and try and sort of do a bit of a potpourri and just give 30 seconds on or a minute on everybody's current projects, but really that would have taken too long. So I'll just introduce all of these people. We've got Justin here, who's into physical anthropology. That's looking at essentially human evolution. Chris Goatley, who's a postdoc. Sorry, Justin's a postdoc too. Chris actually looks at small coral reef fish. We've got Ada, who looks at long-necked dinosaurs. We've got Rex, who looks at kangaroo skull mechanics. John, who looks at locomotion in kangaroos, including extinct giant kangaroos. Theo, who looks at primate skulls. Michael, who looks at one of my favourite animals, the extinct marsupial lion. Emma Doolan, who's looking at hyoid bones in hominids, members of our own family. Leah, who's looking at raptor claws in everything from giant eagles to smaller falcons and kestrels. We've got various other people associated with the lab who are either sort of co-mentoring or co-supervising, or paid members, such as Sarah here, who happens to be married to Justin, just for a bit of trivia there. That might come up in a question later on. Russell, who's looking at, you can't see it here, but trilobites, so we even sink as low as invertebrates. Sarah's looking at primate brains. Marie's looking at bird eggs. We've got a guy looking at the water-land transition. And there's a bunch of other stuff. That's just, I've sort of ran out of space and time. So, what are we doing? Are you going to tell me when I'm close to running out of time? I'm probably halfway through. Right. So the common denominator in all of these projects and all of the stuff that we do is that we use 3D data, typically serial X-ray data or CAT scan data, and computer-based approaches. And we use those to look at relationships between shape and function, typically in evolutionary contexts. So why use this? Why use these sorts of approaches? There's a lot of advantages, but the obvious ones are that they're non-destructive, particularly useful if you're dealing with rare fossil material. Increasingly, it's easy to transfer data and information around the world. So if we've got CT or models of, you know, a dinosaur skull or a fossil human or whatever, we can transfer that or show it to other people. Alternatively, we can get data, raw data that we need from pretty much, you know, any institution in the world, anyone who's prepared to share it with us. And it gives us a bunch, basically it also gives us a bunch of powerful new tools with which to answer big questions in paleoscience and biology. I'll put this here for a reason, but I don't have enough time, so. One of the major tools we use is something called finite element analysis. What is this? Basically, it's a process that's been used for decades now, first developed by the aerospace industry 50 or 60 years ago. Essentially, it's a way of non-destructively crash testing or initially man-made structures. It's a method, a computer-based method that allows us to basically answer or solve very complex three-dimensional models. Now, initially, it was used entirely for you know, man-made structures, whether it's a spaceship or a car, and these are relatively simple geometric shapes. Over the last decade or so, the computer power or increases in computing power have allowed us to look at much more complex shapes, such as biological shapes. And essentially what we do is divide a three-dimensional structure into typically hundreds of thousands or millions of little simple shapes. So we can predict behaviour, mechanical behaviour, look at something like stress or strain in simple shapes and calculate that quite relatively easily. If you then get a whole bunch of these little shapes and stick them together, and get them to talk to each other, you can solve and look at and predict things like stress and strain in much more complicated or much more complex structures, like skulls, etc. So you build your model. So here, for example, is a chimpanzee skull. You can't see it very well here, if at all, but this is made out of 3 million little tetrahedra. We give those tetrahedra mechanical properties. We tell them how stiff they are, for example. Get them to talk to each other through nodes at the points of each of the tetrahedra. Then we start applying forces. In this case, we're looking at skulls, so we want to look at jaw muscles. So we recreate the jaw muscles and calculate the forces, predict the forces. Then we restrain the model in 3D space to simulate a particular kind of behaviour that we're interested in. In this instance, we're looking at biting at the canine teeth, and then we solve it. And this is a model that's been solved for stress. The lighter colours indicate higher stress, the cooler colours indicate low stress. So it allows us to predict things like stress and strain with quite high degrees of precision. So more precisely, what sort of questions can we answer? I'm just going to go sort of historically through a few of the questions we've asked over the years. Here we have the saber-toothed tiger, Smilodon fatalis, very well-known fossil cat. There's a long-running debate over, or has been a long-running debate over how it actually killed its prey. We used FEA here to show that the saber-toothed tiger was actually poorly adapted to bite hard using its jaw muscles. What we did find, though, was that compared to living cats, 2 minutes to go, Jesus. Anyway, we found out it could bite. It wasn't a good biter. Wasn't a good biter, but it had really strong neck muscles. Yep. Another example, we looked at mechanical performance in modern humans and compared them to other members of our family, including living ones, such as chimps and gorillas, as well as some extinct ones, such as, excuse me, Nutcracker Man here. Hang on, I've got to go back. What did we find? Well, we actually found that modern humans have a surprisingly powerful bite, and not only is it a relatively powerful bite, but their skulls are quite well adapted to sustain those powerful forces. Some physical anthropologists didn't particularly like that, but we got it published anyway. Right. Another example, very quickly. This is some work that we've got submitted at the moment. It's a result of a major collaborative effort. Basically, we're asking the question, why did Neanderthals have such long prognathic faces? There are two sort of functional hypotheses that have been out there for the last 100, 150 years or so. One is that they were adapted to bite really hard at their front teeth. The other one was that they were adapted, that this was an adaptation to really cold environments. Basically, that it was all about warming inspired air through the nasal passages. Oh, sorry. I can go backwards and forwards here. First step in a lot of these things, just super quickly, is reconstructing missing material in fossil data. We've got increasingly sophisticated methods for doing this, but obviously I don't have time to talk about it, but it looks funky. We address these two questions using finite element analysis, which I just spoke about before, and another computational approach called computational fluid dynamics, which allowed us to predict things like heat and water loss in the nasal passage of Neanderthals compared to modern humans and another fossil hominid human that we threw in there. We, actually colleagues of mine mostly, developed some neat new ways of integrating shape data with the engineering results to help us interpret the results. And the results were that Neanderthals weren't particularly well adapted to generate high anterior bite forces compared to modern humans. And they were also less, actually a bit more surprisingly to me, less effective at conditioning cold, dry air than modern humans. That was definitely a surprise to me and others. What we did find, though, was that Neanderthals could blow really hard. Basically, they could move a whole lot of air into their lungs and out. That has all sorts of ramifications in terms of the stamina of and sort of the long distance running or whatever capacity of Neanderthals. Suggests that they might have basically been running hotter, had a higher metabolic rate, or both. I'm just going to keep going till you shut me up, James. So.
All right, I'm going to shut you up then.
No, you're not. I'm going to finish this one.
All right.
Just very quickly, there's a whole bunch of other of applications. We can look at model bone in extreme detail, such as we're doing here. We're modelling right down to the trabecular level in some human bones. We're looking at hyoids and how that flows on to work on the hyoid, which is a bone that's very important with respect to speech and the evolution thereof. We do some surgical type stuff. Where's it all going? With advances in computer power, we can now include much larger data sets, solve many more models and bigger models faster. So we can basically ask bigger and bigger questions. And I'll leave it at that. Sorry.
All right, round of applause for Steve, everyone. All right, so Steve, before you go anywhere, essentially the short story is you're getting these skulls and biological bits of things that could have existed millions of years ago, taking scans, making reconstructions, and answering questions about how they ate and how they breathed and how they moved. That's nuts. That's good.
It is nuts, but it's fun.
No, and when I think about paleontology, I think about sitting in a hole with a toothbrush and brushing away dust from stuff, but that's not what paleontology is anymore.
I've got minions that do that.
All right, well, round of applause once more for Stephen. We'll bring up our next speaker, who's Nick Campione. All right, everyone, this is Nick Nick, you're brand new.
Hello, everyone.
You're brand new to Armadale, right?
I am a Canadian paleontologist. So if I sound funny, I'm sorry, eh?
I think I got the funny accents covered, Nick. I'm sorry. So how long have you been in Armadale now?
I've been here for 4 weeks.
All right.
Yeah.
All right. Well, I'm gonna warn you. These Armadale crowds,
They're a hard,
ruthless,
a tough crowd.
unforgiving.
I can see them. They're salivating.
All right. So take it easy on Nick, everyone. I'm gonna hand it over. Thanks, Nick.
Thank you very much. So it's a pleasure to be here. I've never done a Science in the Club before, but it seems like a really good opportunity to sort of expose everybody to the science that's going on in the world around them. And I, for one, have been very curious about the world that I live in. And so it's nice to see so many people who are also equally curious about this. And so my name is Nick Campione, and I am a dinosaur paleontologist, although I work with a variety of different organisms as well. And over the last few years, I've worked a lot on the question of dinosaur body size. How do you attain it? What does it mean? And questions like that I'll try to share a little bit with you today. And so the question that I wanted to start off with actually was, does anybody know what the largest dinosaur is? Let's go with you.
T-Rex.
Okay, that is a very big dinosaur, but not the largest. Yeah. It is a titanosaur, but what kind of titanosaur? Vicious is very big, but the specimen is disappeared. No one knows where it is. Brachiosaurus. These are all very big dinosaurs. Okay. Oh.
Argentinosaurus.
Ah. So for 25 years, Argentinosaurus has been the largest dinosaur that we know of. And coming in at a whopping 83,000 kilograms, it is definitely supposed to be a very large animal. But this number has always intrigued me, even as a kid looking at these numbers. And so one of the questions that I've had is, how do we know how big of a dinosaur is? It's not like we can put a dinosaur on a scale and just know how heavy it was. We need to do some work to figure that out. And so the story goes back to actually these two characters. This is Henry Fairfield Osborn from the American Museum of Natural History in New York. And next to him is William Gregory. Osborn is known for naming and discovering a dinosaur called Tyrannosaurus rex, which I'm sure you've never heard of. And Gregory was one of his people that worked with him. And Osborn in 1905 asked Gregory to give him an estimate for a dinosaur called Brontosaurus. He based it off of a reconstruction by a paleoartist named Charles Knight, and this is the specimen here at the American Museum of Natural History, and he generated an estimate of about 31,000 kilograms. So much less than what we got for Argentinosaurus. The method was quite straightforward and followed Archimedes' principle, and which essentially comes down to dunking the model in water to determine its volume. And so the process starts with your reconstruction. From that, you dunk it in water to determine the volume of the model. From there, you can scale that model volume up based on measurements of the actual specimen to determine the volume of the dinosaur. You multiply that by some sort of magical number of density in order to determine the body mass of the animal. So this has been one of the long-running methods of estimating body masses in dinosaurs. And as you can see, there are many steps. And so some recent methods using a lot of 3D type information, such as scans from museum mounts, have tried to sort of simplify this approach. And what they've done is, rather than reconstructing specimens physically, they reconstruct them in 3D using computers. And so rather than doing all this arduous process, you go straight from your reconstruction all the way to the dinosaur volume. However, you still are dependent on that reconstruction, and you're still dependent on some estimate of density, which are very difficult to determine in fossils. And if you remember our 83,000 kilogram Argentinosaurus, which is walking here, it suffers from an even bigger problem. And that is that specimen is only known from about 10% of its skeleton. Most of it is what a lot of us would term paleofantasy. It is missing a lot of information. And so perhaps you have heard of this animal, Patagotitan. Anybody heard of Patagotitan? Well, that's because it was presented to us earlier this year. It was just published on, and it is a much more complete dinosaur than Argentinosaurus. And actually, when they've looked at comparable specimens, namely the vertebra, and by the way, that's a silhouette of me next to one of the vertebra of Patagotitan, it is almost always larger than the Argentinosaurus specimens that are available. And what they got as an estimate on a more complete specimen is something between 44,000 and 78,000. Still a massive animal, but not as big as that estimate that had been determined for Argentinosaurus. And so this sort of suggested that maybe the lack of information for Argentinosaurus played a factor in how big this animal actually got. And maybe we wanted it to be bigger than it actually was. So this is currently the reigning champion in terms of the largest dinosaur that we are sure of that we have good data for. But this lack of information, to me, has always been really puzzling and really kind of troubling. And so I forwarded a different approach, that of using scaling relationships. And this is not as fancy as reconstructing the whole animal, but what it's based off is taking a few measurements from living animals and comparing that to the body mass of those living animals, creating a scaling equation, and then using that equation to estimate your fossil data. And so it's as simple as going out, taking the measurements of dinosaurs, tracing it up to the line, and boppity-boopity, you have a body mass estimate. But the method was criticized quite extensively, most notably because we didn't really understand how those relationships differed in living animals. And so a big focus of my research was to go out and collect as much information regarding different limb measurements in living animals and see if I can show the potential of these reconstructions, of these measurements as a predictor of body mass. And I show that, for instance, femur length actually is not a very good predictor of body mass. You can see if you look at the lines here, they're scattered relative to each other, they're in different angles. So that would not be a very good proxy for size. However, if you look at something like circumference, those lines seem to all follow a common trajectory. And so we proposed that this was a pretty good and accurate method for estimating body masses in dinosaurs. And so the advantage of this, of course, is that you don't need to have these complete specimens to create these body mass estimates. You can go out there and collect data on just a few measurements, and then bam, you have a giant data set. And we now have over 500 estimated masses of dinosaurs, and that's the biggest list of masses that has been presented so far. And with this data in hand, we can then start asking other, perhaps more interesting questions, such as, how did dinosaurs get so big? And so for that, we actually traced body size throughout the evolutionary history, the evolutionary tree of dinosaurs. And what we found is two very, very, very interesting patterns. The first is that starting at an animal about the size of a large wallaby, body mass in dinosaurs radiated very quickly. It diversified very quickly. It went in different directions, so that within the first 10 to 15 million years, dinosaurs had reached pretty close to the upper and lower bounds of the size that they would have. After that quick radiation, however, the speed at which body size continued to evolve slowed down. And this is a pattern that evolutionary biologists refer to as an adaptive radiation. And we hypothesized that this reflected sort of a rapid early utilization of many different sort of environments and niches early on in the evolutionary history. And that after that early radiation, all those niches were filled up, and so the evolution of body size kind of slowed down. Essentially, dinosaurs had found their groove. But there was one group that did not follow this pattern, and that is the Maniraptoran dinosaurs, which is the bird lineage, and this includes animals like Velociraptor, Oviraptor, and birds. They continued to evolve new types of body size throughout their history. And this included experimenting with very small sizes. And this we've attributed as being critical in sort of the survival of birds across the extinction, whereas other dinosaurs were kind of stuck in these body size ruts and they couldn't sort of diversify into other environments when climate changed very, very quickly at the extinction event. And so with that, we find that actually dinosaurs diversified quickly. Their body size reached maxima early on, But why did dinosaurs get so big? And this is a very tricky question, a very common question. Everybody wants to know why they got so big. But it's a very tricky question, and not one that I'm obviously going to answer completely today, but I'll give you a couple of kind of thoughts that I have regarding this question, or that I and my colleagues have. And so let's first start and assess what does it mean to be big? And so let's look at here, dinosaur, this is body mass on the bottom here. And we'll look at the three major groups of dinosaurs, the sauropods, the long-necked dinosaurs, the ornithischians, the bird-hipped dinosaurs, and the theropods, the meat-eating dinosaurs. And we can plot out the range of body sizes in those groups. And we see that overall, there are very strong similarities in the ranges in the ornithischians and the theropods. The sauropods do, of course, get a bit bigger. However, if we add on the Maniraptorian lineages, including the birds, the theropod lineage goes all the way down to about 1.6 grams, with the bee hummingbird as the reigning smallest dinosaur out there. If we then also look at mammals, living land mammals, so we'll get rid of the whales for now, we see that actually the range of mammal body sizes matches quite closely with that of meat-eating dinosaurs. And if we attack on the largest mammals that have ever lived, we're actually pretty close to the largest ornithischians as well. And so what this sort of showed is that seems to be sort of an upper bound, sort of a limit at around the 10-ton mark, where three very different land-based groups sort of reached and then stopped getting bigger. And so this sort of hinted at some sort of a common limit in the body size evolution of these groups, and it's really only sauropods that somehow were able to break free from this limitation. And so let's delve this almost done. So let's delve into this a little bit more, and let's look at the distribution of body sizes in living living mammals. This is a pretty common curve with a lot of small things, not so many big things. And the shape of this curve is sort of dictated by short-term advantages to get larger. And so you can think of, if you're larger, you're stronger, you can outcompete your neighbors, you can protect yourself against predators. And so there's a sort of a tendency over time that that animals tend to get larger as they go because of this sort of short-term advantages. But these are countered by sort of a long-term constraints, and these could be physiological. And so a hot-blooded animal, can't get too big because it'll have a hard time dissipating heat, but also increases in extinction risk. And this is an important one because large animals, they reproduce slowly, they have smaller populations, and they have less variation for natural selection to sort of act upon them when changes in environment happen. And so this creates sort of a trade-off mechanism between these two that sort of shapes this graph. And so what we suggest is that this sort of trade-off may actually explain that common limit with a lot of groups. But what did sauropods do? And here we suggest that there is, for instance, some evidence to suggest that that sauropods were able to reproduce a lot faster than one would expect for an animal their size. Their clutches have reached about 40 eggs, huge for a very large animal. Think of an elephant, can basically reproduce once every 2 years. These eggs were sort of probably laid in massive mounds, similar to what marine turtles would have done today. And so in doing that, sauropods may have been able to maintain higher populations than would have been expected for a mammal of an equivalent size. And so in that regard, you basically would have had an alleviation of those constraints on a sauropod evolution, so there was no longer that extinction risk, and that it would have extended that sort of short-term advantage for them to get larger. So the answer to why did dinosaurs get so big? Well, it may be as simple as saying, they got big because nothing was stopping them from getting big. And so with that, I do have more that I can talk to some people about.
Nope.
But I will let someone else.
Next time we do this, I'm going to get one of those big red faces gongs, and I'm just going to whack it, and you can't talk anymore. Round of applause for Nick and everyone. And now, Nick, I really liked your description of the unknown dinosaur bones as the paleofantasy stuff, and that's kind of a lot of dinosaur reconstructions is stuff that we don't know.
There is no single dinosaur that's known from 100% everything.
Yeah,
everything is missing something. Even the best dinosaurs are missing something.
I will say, though, I was on Amazon the other day looking for dinosaur books and searched paleofantasy. Not what I expected.
Well, you can fix that in your browser if you want.
Let's thank Nick one more time. We'll bring out our next speaker, who's John Patterson. All right, John, as a fellow invertebrate biologist, I'm gonna assume that you're gonna bring it home and convince everyone that these little critters are much cooler than the big, bony, bitey things we've been hearing about today.
Yeah, I've got a challenge ahead of me because I don't work on big vertebrates.
You work on
like
much
dinosaurs.
cooler things. I'll leave you to it. Fair
Thanks,
enough.
mate. Cheers. So, as my title slide is telling you, I actually work on really, really old stuff. So, as I said, I don't work on dinosaurs, I don't work on saber-toothed cats or anything like that, like the other guys do. I work on probably what's best described as really ancient seafood, okay? So what I'm interested in is the origins of a lot of the animals we've been talking about already tonight. So, there's dinosaurs. In paleontological terms, they're pretty recent, actually. Fossils go way, way back, billions of years. I don't quite go that far back. I go back about 500 million years, so just over 500 million years, so 500 with 6 zeros at the end. And I'm interested in what was living in the oceans at the time. So a lot of what you can see in this slide actually has its origins around about 500 million years ago. Particularly things like arthropods. So arthropods include crustaceans and spiders and centipedes and insects and so on. But other things like mollusks, so squid, sorry, I know it's okay, it hasn't worked, that's good. Oysters and so on. These are all appearing for the first time just over 500 million years ago. And this particular point in Earth history is what we call the Cambrian period. So geologic periods, some you might have heard of the Jurassic period or the Triassic period or the Cretaceous. The Cambrian periods are much earlier than those, so we're talking way before the time of the dinosaurs. And in my biased opinion, I think the Cambrian period's probably one of, if not the most important evolutionary events in the history of animal life. Because really, this is when animal life kicked off, okay? It's the beginning. And that happened around about 541 million years ago. And what do we see at this time? We see most of the animal groups that are familiar to us today appearing, at least the ones that lived in the oceans. So your crustaceans and so on and so forth. Animals started to work out that they could burrow into sediment. That was a fairly novel thing. We see animal reefs appearing for the first time, even complex ecological interactions, so things eating each other. Okay, this is all very new at around this time, and the list goes on and on and on. So it's what's been dubbed evolution's Big Bang, at least in terms of animals appearing on the planet. Now, I've been lucky enough for the past 10 years now to be working on a very special fossil site on Kangaroo Island in South Australia. And the fossil deposit is called the Emu Bay Shale, and funnily enough, it's near Emu Bay on the north coast of Kangaroo Island. And this is not your ordinary fossil deposit, okay? So Nick's been talking about dinosaurs. Normally when you go out and you find dinosaurs, you'll just find dinosaur bone. It's very hard to find dinosaur skin or any other soft tissue that the dinosaur might have had. It's the same with invertebrate fossils. So a favourite one of mine is a trilobite here, which I'm holding, and trilobites are actually quite common as fossils, and the reason for that is because they have a mineralised skeleton. So trilobites are made of a mineral called calcite, or it's calcium carbonate, and because it's made of that mineral, it preserves readily. But what you wouldn't expect to see in a fossil deposit are all the soft parts of a trilobite, its gut tract or its walking legs, because they're all soft, they're not mineralised. But this special fossil site in Kangaroo Island, we get all the soft, squishy stuff too. So we get things like worms preserved, and this is another type of arthropod here, probably related to a crustacean. And you can see these funny little zipper-like structures that would have run down about that part of the body there. They're actually the digestive glands of this thing. So we're getting very exceptional preservation at this site, which is telling us a lot more than any other fossil deposit normally can. So we can learn a lot more about the anatomy, we can reconstruct the community that was living in the ocean at the time, and it tells us a lot about how evolution's working at this particular point in Earth history. Another thing to remember is the age of this fossil deposit, so it's 514 million, that is. And sometimes we find really weird fossils. Now I'm going to give you 3 goes at this. Hands up if you think you know what this fossil is. Yeah.
It's some sort of mollusk like an oyster.
A mollusk? No, it's not a mollusk. That's one down. Yep.
Like a crab or a lobster or something?
No, it's not a crab or a lobster. No, you don't count, Nick, sorry. Yeah.
I don't know the name, but it was something with a stalk and something that lived on the ocean floor. It was
With a stalk?
it had a stalk and then the shape that you see.
A crinoid? It's not a crinoid. Now, there was one person in the audience up the back who I will let answer this question. Russell, tell everyone what it is.
It's a beautiful example of fossilized poo.
It's poo. It's really, really old poo, 514 million year old poo. And the reason we know that is because what you're seeing here is a nice log shape, but inside it are the broken up fragments of a trilobite. So on occasion, they're not that common, but we will find fossilised poo, or what we call coprolites. And that in itself is really important because it can tell us about diets, or I was talking to someone about this earlier, the first paleo diet, if you like. Sorry, bad, I know. Don't throw things. So why is this particular fossil deposit important? Well, you're probably already starting to get a good idea of why. But if we look at all the animals that have a hard part, which would normally be preserved, and all the animals that purely have soft parts, there's no hard part whatsoever, in this particular deposit, almost 3/4 of the biota is soft-bodied. So in an ocean setting like this, where you'd expect to see all these little Cambrian critters swimming around, if we take out all the soft-bodied animals that you would normally see in a fossil deposit, in other words, you would typically only see the hard parts, you're left with that. So we're getting a much more comprehensive picture of what's occurring in this particular deposit and what was living there at the time. So we've been working here for the past 10 years now, going there twice a year, so we've had 20 field seasons. We've found lots and lots of specimens. I think there's about 6,000 now registered in the South Australian Museum. And as you can see from some of these reconstructions, we're starting to get animals that are looking familiar to what you would see on your seafood platter, including things like that guy up there, which looks a bit like a prawn. But on occasion, we also find something really spectacular. And it actually relates to this. Now, if you're not sure what you're looking at there, you're looking square on to an insect's eyes, or what we call compound eyes. You can see they're made up of tiny little lenses. And contrary to what Hollywood would make you think, it doesn't see in a mosaic. It sees all of these little lenses act like a pixel on a computer screen. Okay, so it builds the complete picture. And I think it was in 2010, I was the lucky one, just happened to pick up the right rock at the right time, split it open, and I found the eyes of this guy. Now, this is Anomalocaris. It's probably, well, you could consider it the great white shark of the Cambrian period, okay? So this thing grew up to about 1 metre in length, pretty gnarly looking claws on the front that it would have used to grab its prey. But the important thing about finding its eyes is that we can learn a lot more about how this thing was looking for its prey. And what was interesting too is that we'd been finding body parts of this animal for quite some time. We'd find the front claws isolated, or we'd find the body flaps that it used to swim just on their own. But to find the actual pair of eyes of this thing was quite exciting. And we published this about 6 years ago now, I think. And even though the fossil itself doesn't, you're like, it's a couple of orange smudges. That might be the case, but these paddle-shaped structures are the two eyes of this thing. And when you start to zoom in on those eyes, you can start to see, remember that characteristic looking structure that I showed you with the insect? You can see these individual lenses within the compound eyes. And what was even more astounding is that the eyes of this thing, or at least one of the eyes, on the preserved surface has about 16,000 lenses. Now, to put that in perspective, if we look at a trilobite that was living around about the same time, but a fairly simple trilobite, didn't have many lenses, about 84 lenses. If you compare the lenses to pixels, pretty fuzzy image. You compare it to something like a housefly. Okay, houseflies have about 2,800 lenses in each eye. The picture's still a bit pixelated, but you're getting a sense of what's around. Dragonflies have about 28,000 lenses, and you can see that the picture is much clearer. Now, I said 16,000 lenses preserved, but we believe that there's probably more on the other side that's still buried in the rock. And if that's the case, it's got more lenses than a dragonfly, and when it comes to compound eye lenses, dragonflies hold the record. So over 500 million years ago, arthropods had worked out that they can develop these huge, sophisticated eyes for hunting. So on that note, I'm going to finish on time.
All right, round of applause for John. Thanks, John. I actually think that may have been under 10 minutes. Good job. Before you go anywhere, I want to tell us about shale ale.
Oh, okay. Yeah, everyone keeps asking about this. Yep. So this last September just gone was our 20th field season. So it was 10-year anniversary for digging on Kangaroo Island. And one thing we do when we're parched at the end of a day of digging is on the way back to our accommodation, there just happens to be a brewery there. And we pop in and have a couple of schooners or whatever. And it was back in April, the season before, we were thinking, we need to celebrate the 10th anniversary of digging here. And I can't remember whose idea it was now, but it was probably after
You
a few.
can claim credit, that's fine.
It was either me or Russell, I can't remember. That we thought, well, it would be fun to ask the brewer if he can actually make a shale ale and see what ideas he can come up with. And we went to him, pitched the idea, and he said, actually, that's a really good idea. I like that. Can you give me a bucket of shale chips? Because obviously when we're breaking the shale open, there's all kinds of chips just falling to the ground and we just discard them. And so we filled up a bucket and he actually filtered the beer through the Emu Bay Shale. So it really is shale ale and you're essentially drinking trilobites, which I think is quite cool.
So these are pretty common fossils then.
Oh, yeah.
Use them for
Yeah.
brewing.
Yeah.
Right, well, on that note, we're going to take a quick break, go grab a drink, get some fresh air. Let's hear it once more for our speakers. And we're going to start up again in about 10 minutes. So my first question is for Steve. Now, you talked a lot about very fancy science and technology and stuff, but my most pressing question is you named your lab the Fear Lab. Was that your idea?
Sorry?
You named your lab the Fear Lab. Was that your idea? Use the microphone.
Actually, it was sort of kind of the idea of a postdoc of mine, Marie Attard, who basically put it out there on Facebook, I think. Said we want a new name for our lab.
It's a bit of a goldmine of a name.
Yeah, it's
Coming
a
up with
terrific
a good
name.
acronym.
Yeah, no one else has got it.
Good.
And they have to come and see us if they want it. and pay money. Yeah,
So you've trademarked it.
absolutely.
Smart move.
Copyrighted, whatever.
Prepares your students before coming there.
Exactly.
The comment was, it prepares our students for coming to work with Stephen. Right. Now, we have a roaming microphone somewhere. Oh, hands up who has a question for our panel. We have one over here. Remember, there's no such thing as a stupid question. I'm going to ask for clarification on what the hell a tetrahedron is later.
That would have been a good.
My question's for John. How big was the rock that you cracked open that had the eyes in it?
About the size of the palm of my hand.
All right.
Yeah, it was pretty small. Just looked like any other bit of shell that I was cracking open, and it opened it up, and the eyes were peering back at me. It was
You
pretty
won
cool.
the lottery.
Yeah,
So your career is based on dumb luck, is what you're saying?
yeah, pretty much.
All right, good. Next question.
Did you know there were eyes straight away?
Yes. So we'd actually found eyes of a different type of arthropod a couple of years earlier, and so we're on the lookout, so to speak, and these ones. Not the groans. Quiet, Russell. Get out of here. Yeah, so I was on the lookout for that pattern, that hexagonal. It's what they call hexagonal packing of those lenses, and what I wasn't prepared for was the size of them. So, and that's something I didn't say in the talk. Each eye is around about 2.5 to 3 centimeters long. So it's a really big eye.
These are pretty big creatures overall.
Yeah. I mean, Anomalocaris could probably grow to about 1 metre. Some of its younger cousins could probably grow to 2 metres, but the 2 metre cousin was probably a filter feeder, not a big gnarly predator like Anomalocaris.
Yeah. All right. Who's next? We had a question over here somewhere? Yep. Oh, use the microphone.
What's the biggest thing that ever walked the earth?
Oh, I think Nick can cover this one.
That walked the Earth. Well, I guess that would be Patagotitan.
So, how big? What are we talking here?
Patagotitan?
Yeah.
Well, the estimates put it at somewhere between 44 and 78 metric tons.
So, are we still sure that whales are bigger than these dinosaurs?
Oh, whales are definitely bigger than
Yeah,
dinosaurs, but
okay.
whales don't walk the Earth. They swim
Good.
the Earth.
I'll buy that. That's
Well, and it's really only just the one whale. The blue whale
Yeah.
is the largest animal ever.
ever.
Yeah.
Do you think? I mean, we keep discovering more dinosaur fossils. How close are we getting? Might there be a day we see someone beat it?
So this is the slide that I wasn't able to get to.
Oh, what a shame.
What a shame, indeed. And in it, I talk about how the body size of sauropods seems to be linked to temperature. And so if we take that at face value, it would suggest that the largest sauropods were dictated by the temperature around them. If it got warmer, they may have actually gotten even bigger. So we may not actually know what the limit is possible of a living land-walking animal.
And that's despite the fact that warmer temperatures mean warmer or more heat stress on big things.
Right, but if these things were, so again, part of that slide that I wasn't able to get to,
God,
it would suggest that these animals may have actually been cold-blooded. Just the sauropods. Other dinosaurs may have been warm-blooded, but the sauropods may have maintained that sort of cold-blooded nature, and that combined with the fact that they weren't limited by reproduction would have allowed them to get bigger.
All right, good.
Next time I'll plan the talk so that it doesn't go over the limit.
I mean, next time. I think you're coming back. What's
This is for another science in the club.
one more question?
If you could bring back dinosaurs, would you?
Oh,
Well, dinosaurs are among us as birds. They're already here.
but they're little and they're fluffy. And which would you bring back?
I probably would enjoy seeing some hadrosaurs. Those are my favorite group of dinosaur. They seem, relatively docile. I don't know, but it seems like they would be. They don't have sharp teeth, so
Hadrosaurs are
the duck-billed dinosaurs.
yeah. so Ducky from Land Before Time.
Correct.
Good, got it. Somebody asked before what my number one favorite dinosaur was.
And oddly enough, Armadale has 2 world experts on duckbill dinosaurs, myself and Phil Bell, who also works at UNE. And I don't think there's anywhere else in the world that can boast that.
Well, I mean, UNE in general has a really high per capita level of paleo people. This has got to be some sort of record, right?
Yeah.
It is, actually?
Yeah, we're by far the biggest paleo group in the country. So I think we're up, what are we up to now? 12 paleontologists? At least. Something like that.
Southern Hemisphere.
That's
And
like
that-
half
that-
Armadale.
And that's not
God.
including research students. So you include PhDs and masters, and it's huge.
Why is the...
At least 3 more postdocs next year.
Yeah.
Is this a particularly good place for dinosaur work? Or...
Okay, so
Dinosaurs, that's amazing.
actually, Armadale isn't that exciting for fossils, really. So it's unusual that the biggest paleo group in the country is based in Armadale, where there's not a huge abundance of them, of fossils, that is. But I think it's kind of just snowballed. I think it's critical mass. So
It's all down to you guys. You're just great.
as the group has grown, we're getting more people who just want to come and work together. So it's good fun.
All right, any more questions about how good these guys are? One down the front. You finish your drink.
Someone may have said this already, but how long is the Titan?
How to
thing?
go Titan?
Yeah,
thing?
that one.
Oh, I don't remember.
I wrote down
I don't have an answer for you.
37 meters. on
I've
my
only
thing.
been interested in the body mass of these animals. I've never been interested in how long they were.
37 meters. Yeah, that's the answer.
So length is a problem, though, because in general, most of the animal is not preserved. Remember, we talked about that? And so the tail is often not entirely preserved. And so the full length is very difficult to determine for these animals.
So that leads to another question. You talked about how we reconstruct or how we estimate how big they were.
Mm-hmm.
How do we then estimate what they looked like?
So you mean in terms of
If you've
how
only
much-
got bits and pieces of a skeleton, how do you then just rebuild it from those bits and pieces?
No, this is a very good question. It's quite tricky, actually, to kind of figure out. And we base our reconstructions on sort of the animals that we see around us. Everything that we do is biased by what we see around us. And we try to reconstruct the dinosaurs the same way that we would be how we observe the animals. But this brings up some interesting issues. You know, the elephant, if we didn't know what an elephant looked like, would we put a trunk on it? Most likely not. Yeah, so maybe some of these dinosaurs were walking around with giant trunks hanging off their noses, but we don't actually know that. So,
Can I point out something about that?
Use a microphone.
We've got the Asian tapus something, and that's got a trunk. But if elephants didn't exist, we might be able to pick up from the skull shape of them that elephants would have a trunk. So maybe I don't think dinosaurs would have trunks because they don't have the right skeleton skull shape.
would you
Which
agree
is
with that?
kind of what you do,
They
Stephen.
definitely are marked.
Use a microphone.
To a degree, for sure.
Pass it on.
Definitely,
Microphone.
There definitely are features in the skull that we know from looking at living animals, whether it's an elephant, which has got the most extreme kind of trunk, or animals like tapirs, which have relatively small trunks. And those can give us an indication of whether or not an extinct animal actually did have that soft tissue in a trunk. And Australia interestingly, in the Pleistocene, in the Ice Ages, had an animal called Palaecolestes, which we think probably had a tapir-like trunk from looking at its facial
It's
morphology.
schnoz.
Well, we don't have a schnoz,
Don't
but
have a
we do have similar sorts of things to looking at forensic science, looking at rebuilding soft tissue in humans. You can't rebuild a human nose from a skeleton, obviously, but there are indicators because it's cartilage and soft tissue. But you can get basic predictors, so you can get an idea of how. From the size and shape of the nasal aperture, you can get a pretty good indicator of how wide and how long a person's nose was. We've got a lot more data, obviously, when it comes to people and their noses, but we can apply the same general approach for extinct animals. So you're sort of kind of right. But another thing, just quickly, when it comes to reconstructing extinct animals, obviously, the less complete the material is, the more open to interpretation it is, but we can increasingly apply sort of rigorous quantitative approaches to predict that shape based on what we've already got. So we can take, say in our lab, what we do, or have done certainly with some things, is we take a skull, or what's known from the skull of a living species, an extinct species. We take a living species that's the closest in terms of overall shape. We put a whole bunch of landmarks on those two specimens, and then we can warp the shape of the living specimen to fit the known morphology of the extinct specimen and fill in the gaps. And it's a repeatable procedure. So it's more scientific than just like totally making it up. It's repeatable.
Yeah.
The more missing data, obviously, the less certain you are. But there are ways ahead in this. Yeah.
All right, we have a question up the front who's been waiting very patiently.
What is the biggest Spinosaurus?
Oh, that's a very specific question.
And what was his name?
Steve. Steve. The biggest Spinosaurus, well, I guess it'd probably be the one that was discovered. And my estimates of Spinosaurus put it still lighter than T-Rex. So probably around the 6 or 7 ton.
Wait, wasn't Spinosaurus the one out of Jurassic Park 3?
It was.
That was
we
bigger
forget
than
that
T-Rex?
movie though. I've never seen that movie.
Yeah, it was
Yeah.
bigger in Jurassic Park 3.
But if you've not seen, there was a recent paper, well, recent in the last few years, that found more specimens of Spinosaurus. And Spinosaurus may have actually been quite an aquatic animal, the only dinosaur to be perhaps more aquatic than terrestrial. And if their reconstructions are correct, it would have been very difficult for that animal to get around on land. It would have had very small back limbs, very big forelimbs, and it would have not been particularly easy for it to move around. So T-Rex probably would have had a good time eating Spinosaurus.
One question here in the middle. Just talk into it, it's fine.
Yeah, question for Steve, just on how you're saying how much prediction there is in guessing what shape an animal is. Has the theory ever had a test? Like, has anyone ever made a prediction of what this creature from these bones look like? And a bit later, someone stuck up one and thought, holy crap, that's what it really looked like? Or
That's a really good question. Not to the best of my knowledge at this point. Because these approaches, we're talking very recent, really. We're only talking over the last 5 years and probably less over which they've been regularly applied. But that will be the test. That will be one of the tests of it, yes. There are other ways of testing it, but that's the best way, for sure.
One more question. I'm going to go up the back there. You can ask these questions anytime.
We have 2 more.
Well, all right, Kirsty's are 2 more. Yep.
How did you get into dinosaurs? This was sort of all of it.
I guess I'm the dinosaur guy. How old are you?
10.
10, okay. And you like dinosaurs?
Yeah.
And when did you start liking dinosaurs?
Well, for the last 5 years.
5 years. Funny enough, 5 years old was about the age where I started getting into dinosaurs. And what spawned me was actually found an invertebrate fossil. So I found a fossil. My father said, no, it's not a fossil. And then I said, yeah, I'm pretty sure it's a fossil. And then I showed it to someone else who knew more about fossils than my father, who didn't know anything about fossils. And they're like, yeah, it's a fossil. It's a cephalopod. And then I was hooked onto fossils. And then from there, at some point, I don't know when I made the connection between fossils and dinosaurs, but at some point I made that connection. And then I was hooked, and I've been into dinosaurs ever since.
All right, do we have one more question?
We do, sorry, where?
You can ask it, go ahead.
Look, I don't know if I can follow that, because that's a really, like, every paleontologist in this room has probably had the same story. This is one's for Professor Patterson. Look, you showed a really nice poo. It's really gorgeous. And I want to know what made that poo, John. Tell me.
Okay, Russell is asking this question because Russell is my PhD student and he's studying this very topic at the moment. So I'm going to say that's your question to answer, Russell. But based on our discussions and what we know, I would think that it's probably a trilobite, and which is interesting because you might remember when I showed that slide and then I put up the picture of the trilobite next to it, the fragments of the trilobite in that fossilized poo is of the trilobite that I showed, the complete one. And Russell and I think that it's possible that this particular species of trilobite was actually eating its own kind, because it has very specialised legs that are used for crushing shell. And we know that based on looking at modern arthropods. So our working hypothesis is that some trilobites are actually cannibals, which is kind of cool. But that's your job to prove that.
You know you're biasing his PhD, John.
All right.
Yeah.
Well, if you have more questions, these guys are going to be hanging around. I'm sure you can buy them a drink or catch them before they leave.
It sounds like a bit of paleo fantasy to me.
But hopefully from tonight we've seen that paleontology is far from the study of old bones. It's a really progressive field, especially with advances in technology and processing power. It's as relevant now as ever. But if I can bring it full circle, I think it was Slim Dusty who said it best in his song Dinosaur, where he said, yes, the dinosaur was a great big thing, but now it is no more. And we should learn before it's too late from that poor old dinosaur. Guys, thanks for coming to Science in the Club. Let's give it one more time to our presenters, and we'll see you here next year.